Welcome back, everyone. Let's present our next company. It's Bioxytran, Inc. They are an OTCQB company trading under the ticker symbol BIXT. The company is a clinical stage biotech with two revolutionary platform technologies. They have an oral glycovirology drug that recently completed clinical trials and achieved 100% viral reduction in 7 days, a feat only accomplished by one other drug in the past decade. Their drug is a galectin inhibitor that neutralizes the COVID-19 virus and prevents it from entering cells. They also have another platform drug being developed to treat strokes. And due to the molecule's small size, it has the ability to carry oxygen into areas of poor circulation. Please welcome back the CCO of Bioxytran, Sheikh, the creator of the Galectin Challenge and the simplifier of all things biotech. We can't wait to see what you have in store for us this presentation. Welcome back, Mike. Happy to have you back.
Yeah, I'm so pleased to be here and excited to go over my presentation.
All right.
Ask some questions at the end.
The floor is yours. Back when you're ready.
My name is Sheikh. I'm going to give the presentation today. I'm really excited to be here, but what I want to encourage everyone to do is stay to the end. We will be breaking some news, so I want you to stay tuned. These are forward-looking statements. Please go ahead and read them at your leisure. Now, we're going to talk about the elephant in the room when it comes to COVID. And it's the idea and the notion that living with the virus is okay and that we should embrace it. The idea is we should embrace the idea, hey, people are just going to get infected. We should embrace the idea that, you know, look, there's a lot of COVID-19 deaths and that's just part of life. We should embrace other viruses like HIV, RSV, and viral conjunctivitis. Lots of people get it. We should just live with it. And finally, what about long COVID? Yeah, that's okay. I mean, it's a cost of doing business, of keeping the economy open. Look, we believe that these statistics are completely and utterly unacceptable. So as a company, if you're looking at us as a company, understand what drives us. What drives us is we believe that something needs to be done. We reject the idea that living with this virus is okay. We don't like the idea that 1.1 million people died. We don't like the people that 2.1 million people have RSV or the 1.2 million with HIV. What about long COVID? 1 in 5 people have long COVID. We think we can do something about it. My mission statement is we're a clinical stage pharmaceutical company. That means we have people in the clinic trying out our drugs. And it's in the field of glycovirology, hypoxia, and degenerative diseases. Now you'll see the 3 things down there at the bottom. Our focus is going to be on glycovirology. But we're going to break some news here on these other 2 right toward the end. Is COVID really over? No one's masking, no one's testing, no one's distancing. Even the president says, look, it's over. So it's become a polarizing event. So whether you're on one side of the fence or the other, it doesn't matter. We want to take a look at the facts. The CDC is no longer saying, reporting daily infections. They're de-emphasizing it and going to weekly. There's 400 deaths daily, and that's 20 out of 50 states reporting. So it's a reporting issue. The number's obviously higher. There's new immune-invasive variants like XBB and BQ.1.1, and 4 million people have been sidelined by long COVID, and that is a low number. This is the conservative Brookings Institute that we're using. And then 1 in 5 people that get COVID present the signs of long COVID. So I ask you, is COVID really over? As a company, it doesn't matter to us. There's no relevance to our strategy. So some of you might think of us as a COVID company. No, we're much more than a COVID company. And that is the message that I want to bring home. So are you investing in a COVID stock? No. We, prolactin, our platform technology, it's easy to prove our indication. We achieved, and you'll find this out later on, viral elimination in 3 days. 3 days. We believe that we can get a label expansion, and it's got a non-toxic profile, so it's going to be treated favorably with the FDA. So our goal is drug approval as soon as possible. Since we're talking about biotech, and some of you might not be biotech investors, I want to go over and teach you what to look for in a biotech. I call it the M&Ms of biotech. You need management and you need magic. You need both of those things. Now, management is basically, you need the regulatory experience to get successfully through the clinical trials. The magic is the science. Does the science work? You'll see we have a couple articles on this slide so that you can kind of see this isn't something that we're making up. Everyone, when they're looking at biotechs, looks at these criteria. So let's talk about the M of the M&Ms. This is a picture of Dr. Platt. Look at some of his, right underneath his picture, look at some of his accomplishments, 2 textbooks. Okay, I'm going to do a visual aid. This is the textbook. He literally wrote the textbook on how to develop carbohydrate drugs. Right here. Been a bunch of public companies, journal articles, patents, clinical trials, animal experiments. The entire clinical trial process. That's his zone that he's in. And he was the first person to come up with the galectin article. Back in 1993, came up with the first article, and it spurred, you can see in the upper right-hand corner, 4,000 journal articles was the inspiration from his first. I want you to think about it. So we have the scientist that generated this huge body of scientific evidence. And here he is developing solutions to treat some of these things. All these 4,000 journal articles, you know what they say on there? They basically say, we believe using a galectin antagonist can help treat this disease. I mean, it's almost a carbon copy. And this leads me to the next point, or from a last presentation, where I talked about the galectin challenge. You can't do the galectin challenge or you can't find disease indications where you can type galectin 3 and you're not going to come up with journal articles. Here's some more about our team. I'll just highlight myself so you know who's given the presentation. I was a financial consultant, research analyst. I've been in the life science sectors. My strength is taking these hard to understand concepts and just distilling them down so the investors can understand, what does this really mean? This is a technology overview of 2 of our platform technologies, the glycovirology platform and then the hypoxia and degenerative diseases platform. For me to really get, or to give you, the investors, a real understanding of how massive these platforms are, I got to go to the next slide. Galectin 3, I'm reading the title, Galectin 3, one molecule for an alphabet of diseases. Look at what we have here. So this is a peer-reviewed journal article that basically says the galectin target is huge. It's massive. For every single letter of the alphabet, there's a disease indication. And we can look at them, like I'll just pick on the one here, NASH. That's just a small little indication. I mean, this is what we're eventually going to expand to. This is our roadmap of the future. This slide. Glycovirology, it's a new word that we've coined. What does that mean? Glycovirology, very simply put, is the study of how carbohydrates interact with viruses. Our platform is a galectin antagonist. It's versatile. We don't care what variant the person has. We believe we'll be able to treat it because we go to the conserved site. That means it doesn't matter what the variant is. It's tested. A little spoiler alert, we'll talk about our results. We eliminated the viral load to undetectable levels in 7 days. So we have a phase 2 study that we're going to go over shortly. There's no expected limitations. You'll see there the little kid to the old man. We could even put a pregnant person in there. There's no limitations using our molecule. It's efficient. It eliminates contagion. What does that mean? You get the drug, and then you stop spreading it, or after a certain period of time, you stop spreading it to people in your family. So what we are in a nutshell is we're the first line defense against all mutation of coronaviruses. So let me distill that down. So what happens is you get your test, you look at it, you say, ooh, I'm positive. You start taking some medicine, you start feeling better, and then you take a test 3 days later, and odds are you're better. Let's move on to some clinical trial results. I'm going to gloss over this very quickly. This was our first trial, and we did achieve statistical relevance. The number's not on there, but I'll just tell you what it was. It was a p-value of 0.029 for those scientific ones in the audience. What we're hoping to accomplish, I'll look at the left chart. We're hoping that those viral load lines dive below the red line, which is the undetectable line. Let's now look at some blinded data. And what is blinded data? Blinded data means all these lines represent viral curves, if you would. They're actually CT values. But I'm going to just call them viral curves so you kind of understand and you're like, why are they going up? But there's an inverse relationship. But what you want to do is there's a red line there. You want to get above that red line as quick as possible, and time is on the bottom chart of it. So you start at time 0, and then you want to get above that red line as quick as possible. And these represent both groups of people. It represents the placebo group and people in the active arm. So when you look at it, do you see any grouping? That's the question. I'm going to show you the next slide. So take a look at that, see if you see any sort of grouping, and then I'm going to give you the answer. There's 2 groups of people. One is placebo, and one is taking the active drug. The people in green, they took the active drug. And what you see here is they get above that red line very quickly. Most of the people, 88%, as a matter of fact, are above that line in 3 days. And then you see everyone, 100% of the people resolved by day 7. But what's interesting is everyone else was above the line. If you look at the chart or the line, they cross right at the 4.5 point. There's another slide over here that really, not slide, I meant the chart on the right, that really makes things simple for you. You have this line of demarcation. It's called an endpoint. It's you're better or you're not better, right? If you're below the green line, guess what? You're sick. If you're above the green line, you're better. So these charts show you how many people got better and how quickly they got better. It's like, do you want to be the blue line or do you want to be the red line? I feel like I'm in a movie of The Matrix or something. You obviously want to be the blue line, and that was the treated group. All right, this is visual aids for no-brainer. Another thing that comes up is the elimination of rebounds. One of the things that happened with Paxlovid is a lot of people got these viral rebounds, and they got it by day 10. And you can see we went out to day 14, and there was no evidence of rebounds. We do not believe we'll see rebounds. How does it work? This graphic depicts how the virus enters the cell, and our galectin antagonist binds to the little spikes proteins. And when it binds the spike proteins, it prevents it from entering. It needs the spike proteins to enter the cell. So if they're blocked in some way by either a galectin antagonist or perhaps an antibody, you probably heard of that, then it prevents the virus from entering the cell. So anyhow, this is our theoretical mechanism, is that we block it and the liver filters everything out. We believe we have an end-to-end solution. We have, the key takeaways from this slide is we have an oral and we have an intravenous solution. And we believe that we can impact coronavirus against all stages of the disease, whether it's from the beginning stage or to the end fibrotic stage or long hauler stage. We're going to break some news here on our glycovirology development platform. And you'll see we upgraded it. We're basically doing another phase 2, but we finished the first, and I showed you the clinical trial results from it. The next thing on the slide is you'll see the prolactin I and prolactin F, and what you'll see there is we're going to be doing some work with that soon. We haven't figured out if we're going to do one or both. And then another little breaking highlight here is the prolactin X, other viral indications. You can kind of see they're in progress for influenza, HSV, and conjunctivitis. I like this slide because it really explains why we're different. You have all these approaches, and there's probably 10,000 clinical trials. And what they're trying to do is they're trying to squeeze that immune system, and they're trying to get it to work smarter, stronger, and faster to clear the virus. That's what they want to do. So we took a different approach, and I believe that we're the only ones using this approach. And what we did is we said, how about we just how about we attract to the spike protein using our molecule and then clear it? That's essentially how we are different. So we're not necessarily utilizing and need the immune system. I mean, we're happy that it's there, but it's not that we need it to spool up. And that's why our results are so dramatic and so quick. You have to say that there's competition. We got the big guys here, the 5 billion in sales, 20 billion in sales. The key takeaways is who our competition is. You can read the names over there, but the bottom, look on the right-hand side, you'll see phase 2, phase 3. We're in the same stage of development. I don't know how to come across the right or wrong way, but are we really competing against Paxlovid and Molnupiravir and these guys? No, not really. We're in our own class. They're going after people with underlying medical conditions. We're nowhere near that. We're taking everybody in our trial. It's a major sea change and a major way to look at things. Now, I'm focusing on the other platform, and I want to leave some time for a Q&A, but I got to take a couple minutes to highlight why this is so exciting, what we're doing here, and why we're breaking some news here. This moves me on to BIXT25. It's an oxygen bridge molecule. So this molecule carries oxygen, and what you'll see is the little blue bubbles in there, and that represents, oxygen. Now, it's 5,000 times smaller than a blood cell. Now, that blood cell's to scale. That's not 5,000 times smaller, but in order for me to show you how it moves through, we have to like expand it out a little bit so you can see that. But it easily passes through the blockage, and it continues to oxygenate. So the plasma is still flowing to these blocked areas. And we really think that this is amazing. And it says the last line there, time reduction of average time to needle by 90%. That's for stroke. So our idea and our concept of why we're so excited about this is, ooh, you have a stroke, the paramedic goes there, and I mean, this is a reenactment, if you would. The paramedic goes there and they give this drug, and then all of a sudden the person gets better. I mean, that's the concept and idea behind what we have here. I'm not making any claims that this does anything, but I wanted to kind of give you a feel and flavor what we hope to accomplish. Now, how does it work? We take, you'll see the graphic with the blood cell. We take the blood cell, we pulverize it, and we end up with hemoglobin, right? There's the heme, and then there's the globin. We want the heme part, and that's that the last molecule there. It's basically an iron molecule that carries oxygen. Now, by itself, that could be toxic. But when we stabilize it with our copolymer, now it's an oxygen carrier. Now, the breaking news is right underneath there. Let me read it to you. So laboratory production line and first batch GLP. So we're finally making the molecule, and we're hopefully going to use this molecule and go into preclinical trials. But this is not our focus, but I wanted to kind of share like the real essence of our company. These are disease indications. Not going to spend too much time on it. Again, we're in the early stages. Commercialization. This is our clinical trial strategy. There's some updates here. So again, some more breaking news. You'll see we're going to be starting a phase 2, phase 3 very soon. And then I and F, we're going to be doing the 1-2s. And then I just broke the news to you about the BIXT 25. So this is our new revised clinical trial strategy. The key highlight here is the asterisk item. You know, we spent $5.1 million to get to this point. That's what I want to highlight. We have some intellectual property, but as a management team, we are waiting to the last possible moment to file some of this intellectual property so that we get the maximum 20 years on it. Now, our strategy, it's licensing, partnership, and then divesting the product line. We're going to be selling this off to big pharma. Our job is to develop the drugs, not sell them. But there was some news that we had earlier today. We should get to that at the end. Here are some comparables, and all these companies were in the same stage of development as we are right now. And look at some of the price tags: 425, 350, 580 plus royalties. These guys were also on our competitive matrix. So what the idea here is, look at this number and then look at our market cap of 50 million. Look at this number and our market cap of 50 million. And then you decide what the right number is going to be. We're obviously going to go after big pharma. And the idea behind this slide is, look, all the indications that we have overlap with almost all big pharma indications. And that pretty much concludes my presentation. So I'm happy to take some Q&A.
Great job. Okay, we do have some questions for you. With so many positive catalysts like your perfect clinical trial results in COVID, why is the stock down so much, Mike?
You know, I just hit on this. You know, for this stage of the development, the stock, it's very undervalued. And if you noticed the slide that I just talked about, like the comparable slide, we should be in the mid hundreds to millions, not sitting at a $50 million market cap. Now, I'm going to give a little bit of honesty here. Starting in June, we were operating on the premise that some of our long-term stockholders weren't going to be sellers. We had a thin float. So what we thought was going to happen was something that's called price discovery. The stock's going to find an equilibrium value and go to there. But then September happened. And in that time frame, we noticed what's going on here. It just seemed like there was something that just didn't seem right. And so what we did was we pulled a DTC report. Now, when we pulled this report, we saw something. We're like, ooh, it looks like we have one seller. The good news is we kind of knew who it was. The bad news is that he was selling. Now, we don't necessarily know the motivation of the seller, but, we can speculate on it. This based on the investor's age, who we knew, it's probably an estate planning thing. But over the past 6 months, what I can tell investors is that they've been responsible for 47.5% of all sales. We crunched the numbers. We know what it is. Now, that's a lot of shares, and it's not something that we anticipated in our plan. But I want to be very transparent with our investors that we believe that he's close to the end of his selling, and we believe that when we announce some, when we announced a lot of people saw that when we announced our groundbreaking clinical trials, on par with Gilead back in 2013, we thought things were going to change, and it just kind of ran into a brick wall. But we believe there's some other catalysts that will help create a little bit of liquidity and allow the stock to once again get into that stage where it's seeking the price discovery.
Okay, that makes sense. And speaking of this perfect clinical trial, so are these types of results normal? Is this too good to be true?
True. The results aren't normal. I got to be candid with you. When I got these results, I looked at them and I was like, man, I got to do some research on this. This is unusual. And then my research took me back to 2013 with Gilead. So I only found one trial in the past decade that had similar type of responders rate of 100% responders rate. Gilead had 21 out of 21 responders when they did their Harvoni trial. Now, Gilead at the time was a little bit of a one-trick pony. I'm putting on my analyst hat there. They were around in, they had the HIV franchise, of course, and then they added this. So, the addition of this, their market cap went from 80 billion to 160 billion. So as an analyst, I could argue, what's something like this worth? Well, maybe 80 billion. I don't know. That's the only data point that we have. Now, I'm not saying that our drug is worth 80 billion, but you have to think what we are worth is maybe some tiny, tiny discount of that. Okay. These types of results that we got, they're rare. And it's rare when you consider that there's probably been well over 10,000 clinical trials conducted over the past decade. Now, let me talk about this. Is this too good to be true? I don't think it is, but I get that people are skeptical out there. Right now, our results are in a preprint format. That means we haven't completed our peer review, but we're really confident that that's going to happen. And we're going to pass our peer review. And then we think that's going to be a major, major value inflection point once a peer review article is reviewed. Now, I also want to take the time to explain that we believe that this really reduces the risks of investing in our company because we've had 2 clinical trials with statistically significant results.
That sounds good. It sounds like the proof is in the pudding. So when will these clinical trials start? And we'll talk a little bit about the hypoxia drug. There's a term that's been batted around. It's called shovel-ready project. So I'm going to pick on that. We actually have a shovel-ready project, if you can call drug development that. We've identified the sites, the drug's being manufactured, and we believe that we can enroll people quickly.
that means this month. But the thing is, I got to put the caveat, it's ultimately about capital and access to capital. And that's where we need you, the investors, to realize what we have here and, help us with the price appreciation. Now, as far as the hypoxia drug is concerned, look, that's dependent upon capital. We really need to focus on getting the COVID-19 drug across the finish line and the approval into the approval square.
And so your company developed a process to create an antiviral drug that may work on other viruses like influenza. That's pretty prevalent right now. So how long will it take for you to develop another indication?
We touched on this basically on our the last preprint, but I'm going to try to recap it. This is the first step in basically expanding our pipeline. We are going to do some RNA sequencing of the viruses. So we're going to be checking the sequence. Now, what we're looking for is a structure called the galectin fold. And once we find that, then what we do is we apply our theory of our knowledge of how the molecules interact and go into that structure, right? And we figure out theoretically if we have a solution. Now, once that theory is done, and that's how we started the first one. It was all theory. And once we do that, then we're going to do in vitro testing and NMR testing. And the NMR is basically nuclear magnetic resonance imaging. It's basically you put the molecules in a chamber and you look for the exciting of atoms and you say, there's a bond, and it maps it. Now, in the eyes of the FDA, we believe doing this process will qualify as kind of like a mechanism of action and allow us to move quickly to develop additional indications and get them into the clinic really fast. So the takeaway out of this is we have a way to get drugs quickly to market.
That's a great way to end, because we do have to move on to our next presenter. Mike, we've got so many questions for you. We're going to send them to you so you can answer on your own time, but that just means you got to come back in 2023, present and give us some more good updates. Thank you for your hard work and your intelligence in this really important environment that you're in.
All right. Thank you so much. And thank you all the investors that came out and saw us today. We appreciate your support.
All right. Thank you, Mike. Happy New Year. We'll see you in 2023.
Sounds good.
Okay, everyone, stay with us. We'll be right back.